Okay, good evening. Can you all hear me well in the audience there and Zoom probably well as well. Okay. Yeah, glad to be back. I think I spoke to your group probably 3 years ago, so maybe some of you have not seen me before. So, so my program here at Oregon State University focuses on very broadly on honeybee health, nutrition, and pollination. So we do a lot of research projects. But today I'm not gonna bore you with all the research projects. I might show in between some research, but I just wanted to make you think about your beekeeping practices and understand a little bit more about Varroa biology. We talk a lot about honeybee biology, but probably you don't get to hear about Varroa biology. So I'm going to focus a little bit more on that. So, and before I do that, Mike, I just wanted to know how many of the audience here are new beekeepers. They just started in the last year or two. I see many of there. I can see if you can raise your hands if you're a new beekeeper, and those in the Zoom also, if you can.
Looks like a good 6 or 7.
6 or 7. Okay. Okay, then I'll keep you guys in my view as well, so that yeah. So other veterans probably can take a nap. I'll try to focus on the new beekeepers here in the audience. But yeah, if you think it's too overwhelming when I ask you, I have a lot of questions to ask as well to make you think. So don't worry. This will be very simple questions, and don't, and even those of you that are really experienced veterans here in the audience, probably you might not have seriously thought about some of those questions I'm going to ask about the Varroa especially. So let's have a fun discussion as well. So yeah, you can ask me some questions if you really can't wait until the end. I can take it, but again, as I was joking with Mike. I'm notorious. I take my time, like I spend a lot of time on my talk, and then if you have too many questions, then we may have to be here until midnight. So, but I'll try to respect your time. Okay, so let's start with. So this is what I'm gonna do. Go over Varroa biology, ask you some questions, make you think about the biology a little bit. I'll show you some studies as well in between that we have been doing at OSU for the last few years as well. Yeah, before I start, let me see. This thing is- Okay, these are some of my staff that have been working at the lab for the last 15 years. So I've been at OSU almost 16 years plus now. So I just want to acknowledge their support. So you'll see a lot of people here. Some of them have left and taken other positions in apiculture or some other industry as well. Some of them are in California as well, in your state. Okay, so so why I'm focusing on Varroa and nutrition here mostly is especially for the new beekeepers in the audience. I always say these are the two important ones that at least have something that you can do to help your bees as well. And I know not everything you can do, but at least there are a few things that you can do in terms of Varroa management and also in terms of nutrition management so that your colony has a higher chance of success. That's what I put here in the slide. You see Varroa and nutrition, I always say if somehow you take care of those two elements, I think probably there is a 90% chance that your hive might thrive. Yeah, I'll start with Varroa life cycle here, especially for the new beekeepers. But again, those of you are very experienced as well. I think it's not bad always a waste of time for going over and refreshing your knowledge about Varroa life cycle because sometimes we tend not to think too much about them. So usually there are two phases, right? You probably new beekeepers might have heard there is a phoretic phase and then there is a reproductive phase. So phoretic phase is when you see those mites on the bee, like you see in this image here. On the left, you are seeing a few mites on the bee. So that's easy, right? You can do a raw sampling. You can do a alcohol wash or a sugar shake or something that you like. And then you can find what your mite threshold is in the hive, right? Like 2%, 5%, whatever the number is. But you see on the right side where I have shown you a frame with brood. If I ask you right now, I know you probably don't have too much brood in your hives at this point, but if this was in June or July, if I ask you how many mites are in those cells, would you be able to tell me? I don't think so, right? Because these are inside the cells, and so that's a part where it's hard. It's really hard to tell how many mites are there, and we'll talk about that in a second. I'll come back to that again. So let me briefly describe the mite cycle, and then I'll show you an animation as well. So the female mites, which are ready, that are already mated, right? And then they are ready to go into the cells to lay eggs. So they'll try to find a larva, which is about 5 to 5.5 days old, and they can smell them, right? Technically, they're not gonna go, they go around the cells to inspect, but they are using the pheromone cues. So they are using this larval cue that is coming from the larva, how old that larva is. So they can find out if it's about 5.5 days or five, and at least they are ready to be capped in the next 12 hours or so. That's what I say here. Female might enter a cell with a larva about 12 hours prior to cell capping. And then they go all the way down and then hide in the brood food that is lying there for the larva. Right? So once the larval food is consumed and then the cell is capped, then they start their business, right? That's how they come and they start feeding on the larva, and then they start laying eggs after that. So the first egg is laid about 2.5 to 3 days after the cell capping. And then subsequently, every 30 hours, there'll be another egg laid. And the first egg that she lays is always a male. And then the subsequent eggs, she might lay five, six, seven, depending on whether it's a drone cell or a worker cell. So that's basically the biology, right? And then the male and the first female, the mites that emerge, they will mate, and then that's how the process goes on inside that. So if you didn't understand anything that I said, then please watch this animation. I think it probably walks you through the phases of this life cycle. Mike, is it running okay, the video on your side? I know sometimes it's choppy.
Yeah, it looks okay today. Thank you.
Okay, thanks. So here is a mite that got in, right? She hides all the way in the bottom so that the bees are not able to remove her, and now she is laying eggs. And then the life cycle of the bee is also going on, right, along with it. So the bee is also progressing in its life cycle. And so mites are not insects. So we use different terms. So we don't call them larvae. So we call them deuteronyms, protonyms. Those are different terms we use. So here is a bee that was inside as a larva. Now she has fully developed and has hatched out of those cells, right? She has chewed the cells and come out. So now you see there is a mother mite that went in, and then she has come up with one offspring baby, right? And sometimes you may get two of those if you have a drone cell. And then again, think about the drone life cycle, right? Drones have 3 more days to go. So then there is a more reproductive ability for these mites to breed inside. So you may get two mites that come out of those cells. So that's basically the life cycle of a mite is, right? So here I have some questions for you. You can unmute, or you can, in the audience, you can yell the answers if you know. What is the lifespan of a Varroa mite? In the Zoom audience, you can unmute yourself and just tell me if you
40
know.
days.
40 days. Wow. You are all, did you read somewhere or you have experience doing a research and you found
No,
out, or?
I just read a lot of books.
Okay, great. Yeah, no, that's a very good answer. Yeah. Uh, so again, so if I ask you, Ella, right, your name? Yeah.
Yeah.
So if I ask you, what's the lifespan of the mite? I know you guys live in California where you don't have technically a winter, but if suppose you are living in Minnesota, and if those bees were in a winter mode for almost 4, 5, 6 months, whenever the spring comes, so those mites are surviving almost 5 to 6 months, right? On the bee.
I guess,
Yeah.
yes.
So your answer is perfect for a regular brood rearing season, right? In July, August, or April, May, the mites are supposed to survive around that. Again, exactly not 40. Average is 40. I think that's a good number to focus on. Uh, but then again, so if I ask this question or someone asks you in the future, you guys can ask, what time are you talking about, right? So it depends. If we are talking in winter, they may be surviving 4 or 5 months. But if it's in summer and spring, probably 40 days. And the reason being, they're so vigorously reproducing, right? They're doing their reproduction like the cycle crazily. So there is a lot of wear and tear on their bodies, and that's how their lifespan is a little lower at that time of the year. Whereas if they are doing that in winter, they're not reproducing, right? They're just being on the bees. They're feeding on the abdominal fat bodies, a little bit hemolymph, and they're just trying to survive until the brood becomes available. for them to reproduce. So that's basically the lifespan of a Varroa mite. And then the next question is, how long can Varroa survive without the host, that is your honeybee? Has anyone tried?
They cannot is the answer from the room.
Oh, they cannot. Okay. So they cannot means like even like 5 minutes?
Shrugs.
Okay. Good commentary. Yeah, thank you. So, no, this is something I just want you to think, because these questions sometimes they may not look very important for you in a beekeeping, but I think they do have some value. So Varroa mites technique, I know it's hard to do. So we have done some studies for some other things. So 1 year I had some mites on my where I am talking right now. This is my office, and so I had some petri dishes with mites from different colonies, and so we were watching how long they can survive without the host. And max, I would see there were a couple of mites that survived almost 48 hours. That means 2 days. And then the rest of them, like 99% of them, they died within probably 7 to 8 hours. So those mites cannot survive without their host. So I know the reason I'm asking is some people think, oh, they will fall on the ground, and so yeah, they are gone. If you don't have if you have a screen bottom board or something, once they fall onto the ground, and they probably have only few hours left somehow to latch onto a bee or something to come in. Otherwise, they'll be dead. So those techniques are very effective. If you have screen bottom boards, if they fall down through the screen onto the ground, yeah. And for those new beekeepers, these mites are, we call them obligate parasites. They don't have any other host. Honeybee is their only host. So without honeybees, they can't survive. So they have to be on the adult honeybee or inside those brood cells reproducing on the larva. So, or the pupal stages. So that's how it is. So that's one. Then the next question is, do Varroa enter another larval cell for reproduction as soon as they come out of their cell? So I showed you that animation, right? The mother came with a baby. What do you think? That those mites will go immediately into another cell to reproduce, or would they wait for a little bit?
Wait a little bit,
Okay. How long?
maybe day or two.
Day or two? Okay. And these are, again, there's no real good answer. These are hard to do studies, right? As a beekeeper, think about it. Like how can you do these studies? So you have to paint a mite, and then you don't know where that mite went, which cell she went, right? So these are hard to do studies. But I think some studies were done in 80s and 90s, not too many, maybe a couple of studies that I'm aware of, that they have documented these things. So yeah, so they can. So the reason being, why would they not? I mean, they have done some studies where they have taken the mite and immediately put into another cell in the lab setting, and they were still successful. But in general, what happens is those mites do not go into a cell directly, because they need to recuperate their energy back, right? Their physiology has to change again, because they were in that cell reproducing for a while. So they are having the lack of energy now, right? So they have to restore their energy, because lack of a good term, I'm just using some vague term as energy here. So they have to feed on those bees again, on the abdominal fat bodies, and then they go. So on average, at least people have documented, they take about 4 to 5 days to get into another cell. And it might be a little more longer for the baby that came out, right? The baby is very new, so she has to still take maybe a week or 10 days. But the adult, that the female that went in before, the mother, she can go as soon as 4 days or so if she wants to. Again, this might be useful information, right? Sometimes when you use oxalic, I don't know how many of you use oxalic vapors. I know there is a Varoxan, which is a strip now people are using, but if some of you use oxalic vapor, that could be another thing to keep in mind is so you can hit your Varroa with the oxalic acid vapors every 5 days, or even some people go 7 days, but five would be an optimal thing. Keeping this point in view that they take about 5 days to go back into a cell. So if you're eating 3 times, 5 days apart, probably you can get rid of probably most of the mites in your hive. So again, and in the animation I showed you, right? So the first female, I told you that the first mite that comes out, the egg that hatches is a male, and then he mates with all the siblings there, right? The other female sisters. So that's technically an inbreeding going on. And we all know that inbreeding is not good, right? Then why do you think the Varroa has been very successful with this strategy? Have you ever thought about that? Am I confusing you too much? Okay. So again, something to ponder on. In breeding, again, there is a haplodiploid system like your honeybees, right? Honeybees are haplodiploid. New beekeepers, when I say haplodiploid, means the males, the drones are haploid. They have one set of chromosomes, and the females, the worker and the queens, are called diploid or diploid, however you pronounce it. So they have two sets of chromosomes. They are like us, right? We are all diploid. We get half of our genes from mom and half from the dad. So we're diploid individuals. We have two sets of chromosomes. Whereas the haploid, the males, they have only one set of chromosomes. So recently I've seen some studies that they are saying, no, Varroa are not haplodiploid there. So Varroa are also haplodiploid. Your honeybee is a haplodiploid. And until now, we also thought Varroa is also a haplodiploid individual. That means the males are haploid, females are diploid. But looks like that's changing. I've seen a couple of papers, but again, we don't know if they are gonna stand. So maybe some people have to really work on those again to prove that they really are not haplodiploid, but we'll see. So we think because of that haplodiploid system, somehow you cannot get those mutations as in a diploid organism like us. Inbreeding is bad, but sometimes in haplodiploid systems, it may not be as deleterious. Again, I will not waste too much time on this here, but if you're interested, you can email me and we can have another discussion at some other point of time. And the last question on Varroa I'm going to ask is, so I said it's all inbreeding going on, right? So to increase their genetic diversity, what do you think? Is there an opportunity for the Varroa to increase its genetic diversity? Wow, great question. Great answer. Yeah. Yeah, sometimes we don't tend to think about that, right? So yeah, sometimes what happens when you have less brood in your colony and you have too many mites, they're competing for the same larval, whatever 200, 300 larvae you have. So sometimes you will see there might be two mites or three mites also can get into the same cell. And when they produce their younger ones, the males and females, then there is an opportunity that the male from this BA may mate with the female offspring from the mite B, right? So there might be exchange of genetics there as well. So there is some, it's not like strictly inbreeding going on, right? So there is an opportunity for exchanging genetics as well in this case. So that's probably is another reason why it is not a total disaster for them in those colonies. Okay, great answers and enthusiasm. So let me go to the So for the new beekeepers again, yeah, I apologize if I'm not focusing on the experienced ones. I know you are all very experienced, so I don't want to spend too much time giving you some basics, but especially the new beekeepers. So you probably heard that they feed on their abdominal fat bodies, right? The bees are having this abdomen where there is a lot of fat bodies stored. And those mites are feeding especially on the lower side. So you see here in this image, you see that cursor where I have the red circle there. So this was a PNA study. Probably some of you have read it, right? And a few years ago, there was a publication in Proceedings of National Academy of Science, a very prestigious journal, where they published this work showing that the Varroa mostly feed on the fat bodies. Before that, we had this understanding that mostly they feed on hemolymph. Hemolymph is the blood of the honeybee. But don't get me wrong, they still feed. I think that paper is not very clear on that. So sometimes when you read it, you get confused. So the bees are still, the mites are still getting some hemolymph, but majority of it is the fat bodies, right? So that's how they feed here on the bottom of the bee. That's where your mites will be hiding between those segments. That's why it's hard for you to see visually. So don't wait to see mites visually on your bees. You need to do your alcohol wash or sugar shake for monitoring of mites without looking onto the bees. So that's important as a new beekeeper. Don't ever think that you have to see mites on the bee. It's too late if you start seeing mites on the bee. And then you see on the bottom here, the bees have this deforming virus. That's a major lethal virus that Varroa mites transmit to honeybees, and so you don't have to wait until too many of bees are shown in your hives like this type of symptoms. So viruses are the major ones. As beekeepers, sometimes we don't tend to think too much about honeybee viruses. So the mite is already doing the damage, right? The mites are doing a physiological damage to your bees. They are sucking the abdominal fat bodies. They're sucking some hemolymph and causing a physiological damage on your bee. But more than that, they're transmitting these viruses. Again, I'm talking about only deformed wing virus here, but there are a bunch of other viruses, from Israeli acute paralysis virus, there is Kashmir bee virus, there is chronic bee paralysis virus. So many viruses are there. But these are the major ones. The deformed wing virus is the most lethal one, as I said before. And as researchers, we still struggle. Like, I'm a honeybee biologist. I do a lot of experiments with bees in terms of bee health, nutrition, and all, but I'm not a virologist. So there are only few virologists that are working on honeybees. And that's a major bottleneck for our industry and beekeepers like you as well, is we have only a minimal understanding of these viruses at this point. So you guys are all tired of COVID and other viruses we have, right? So, and you probably heard of recombinant viruses. So there is always, I don't know if you understand when I say recombinant, that means they are exchanging genetics, right? So there might be, for example, again, I'll use a very layman terminology here. Suppose there is deforming virus A and deforming virus B, and if half of these genetics is mixed with this, and then you have a hybrid, right, which we call a recombinant. So those recombinant viruses are really hard to understand, and we still don't have a good understanding. We have barely scratched the surface in terms of understanding honeybee viruses. So this was a study that showed that we were all thinking that deforming virus A is the important one, but now they are showing that from 2000 to 2020, deforming virus has gone down and deforming virus B has picked up. So we didn't even know there were two variants within deforming virus. So now there is a deforming virus C and a D as well, which probably people have not studied that well. So again, as a beekeeper, you can't do much with viruses, but I just want you to understand that Varroa control is so critical. If you don't control in time, that's the only thing you can do to control viruses, right? Because viruses are so critical. They are the ones eventually that kill your bees. And then they are all dependent on the virus. Most of the viruses are transmitted by Varroa. So Varroa is a carrier of all these viruses. Most of them, probably 95% of them, are carried by or transmitted via these mites. So mite control is the only thing you can do in a better way so that you can reduce your virus levels as well. But don't get me wrong, Varroa is one thing, but there are other ways to transmit viruses in the hive as well. There are some viruses you can do with prophylaxis, exchange of food between bees. So there is another way, but again, the major mechanism or the major pathway of transmission is the Varroa mites. Okay, here is another quick question for you. I think I showed you these slides when I spoke 3 years ago, but again, I know some of you are new to beekeeping and you probably have not seen this. I'll not spend too much time on this, but have you ever thought about how many mites are there inside the cells at a point of time and how many are on the bees? So what I'm asking you is what percentage of mites are in the capped cells, in these cells here that I'm showing, and how many are on the bees, especially if I ask you in your county or your city, probably in June. Any guesses?
We have guesses D.
D, yeah, D is a good number. I think
And-
someone has, yeah, go ahead.
Anybody else want to hazard a guess?
Oh, I'll hazard B.
Somebody said E.
80-20.
80-20, yeah. So the fun part is there are several correct answers in here. Depending on what time of the year. Again, as Ella was giving me that 40 days thing, that's a great answer. But again, it's a depend. Again, I know I said June, so probably you can get a little more close. But it depends on the size of the hive, right? There are so many other factors. So it's not a great question to ask, but I just want you to think about this. So especially the new beekeepers. Again, there is some a practical application for this as well, understanding this type of information. When you use something like oxalic acid vapors again, right? So you need to understand that there are mites coming every day, right, out of those cells. If your queen is laying 2,000 eggs in a day, and if you think there are 50% mites inside those cells, so every day when a bee hatches, out of those 50%, right, are 50% mites are coming. So if 2,000 bees hatch, there are 1,000 mites coming out along with them. So who will kill those mites? I know there are new beekeepers, they will use oxalic because they know they have been told, and so they will use a vapor and they just think magically all mites are gone because I did a very good treatment with the oxalic vapors. But that's not true, right? Because now what has happened? You treated, so the oxalic works only for a day or day and a half, and after that, the product is gone. So your mites are coming every day, right? There are 1,000 mites coming every day. So who will kill those? So that's the reason you use oxalic every 5 days if you're using as a vapor. I know there are other methods that you guys are using now, but that's one way to do it. So for that type of application, this type of information is really, really useful, right? To know how many mites tentatively are there in the brood and what percentage is on the bees is also important to understand. So we did a quick study long back. I'll just, because of time, I'll not go too much into this, but so this was to estimate the mites inside the cells and on the bees. So we chose a location probably 20 miles away from Corvallis, where I am in Oregon, so that we are not spreading mites around to other beekeepers, because this was a mite breeding experiment, not bee breeding, because we don't want to treat mites, right? We want to ask those mites to reproduce as they can in a more seasonal way along with the bees. So we had 6 colonies for that. You will see 6 colonies is not a small number for this type of studies, because it's a very tedious study. Like, it's a lot of work to do in this one. So we didn't treat mites from March until September. And then we went in September and then killed most of the, all bees. And then we took the brood off from those. So this is how we do it. Sometimes we do in a very messy way. You see here, there is a funnel where we are shaking all those bees, and you see it's still a little cooler in the morning. So you see some bees basking on these students and technicians. And so if you want to see the messy part, how we do this thing, you can see how to kill bees here, right?
Another brood frame.
So we collect all the brood and it goes back to the lab into an into a freezer. And then all the bees will go into a bucket of alcohol, which is underneath that funnel, right? When we shake those bees, they're all falling into a bucket with alcohol.
Thanks.
And they will submerge and die. This student is having fun, so we didn't pay her for that day.
Brush wizard.
Okay, so this bucket full of bees come back to the lab, and then we do subsampling of those bees to find the mites on them. And this is the most tedious part, right? You're looking into each cell to find mites. So we gave students with all those magnifying glasses on their visors, and you see when they pull those larva or pupal stages, you see mites on them. So this is how we quantify them. So yeah, we do about 10 projects in a year. So this was just one for that year, right? So it took them almost a year and a half to finish all those frames because that was not the only thing they were doing. And none of them have sued me so far for loss of vision. So here you see a mite family pulled from just one cell, right? So you see this is a male on the right side here. And this is all same family, right? They're not different mites from different types. And sometimes as a beekeeper, you might get confused, right? On your sticky board, sometimes you may see something like this one on the left corner here, lower corner. So these are not sclerotized. So the exoskeleton is not fully sclerotized yet. So that's why they are pale. So sometimes when the bees are removing debris and along with the exoskeletons in the cell, then sometimes these mites will fall, but they're already dead because they're not fully sclerotized, so they can't survive. So these mites that are fully sclerotized, these are the ones that survive. Okay. So someone had a great answer. So you're pretty close, 56% inside the cells and 44% on the bees. So again, it may not be exactly in your area, but I'm just talking about here in the valley where we are. If you look in September, that's probably an estimate you can have. So again, for the new beekeeper, I just wanted to say about 50% mites are inside the cells. And so keep that in mind when you use a product like oxalic or anything that is existent in the hive only for a small amount of time, then you probably have to repeat the treatment, right? Otherwise, just assuming that one treatment is enough will not do the job. Okay, I am pretty sure you all monitor your mites and use one of these methods that I have here, alcohol wash or sugar shake or sticky board. So here again for new beekeepers, monitoring, I can't overemphasize. It's really, really important to look for mite levels. Mites are the major challenge. I know we have all other problems too, but the mites are probably still the number one. So don't wait on calendar-based treatments like I'll do something on Memorial Day or because your bees and mites don't care about those days. They are growing as the season comes, right? So especially we are a little more temperate here in Oregon than what you guys are. So we have to sometimes look for the brood rearing time. If it doesn't rain in February, March, probably then the queens have already started laying eggs in a bigger number. And then we have to think about the season. We have to treat the hives sooner because the mites have a head start now, right? They started breeding sooner than later. So that's important to look for your brood rearing rather than look at the calendar days. So these are some methods. Probably all new beekeepers are already aware how to check mites for you from your hives. But here is a question for you. So if you use any of those mite monitoring tools, if suppose you use alcohol or sugar shake, so you probably are taking one sample, right, from your bees, either like a 300 bee sample or 100. I don't know how you are doing it. Maybe you're taking a jar and just dropping those bees, or you take 2 or 3 brood frames, look for the queen, and then if the queen isn't there, then you shake all the bees in a tub and then mix them well and take a sample. However you are doing it, my question is, what do you think? You get a good estimate just with one sample that you take of your bees, and then if you get a 3% or whatever number, is that a very reliable number? Have you ever given a thought?
No, it's not reliable
Yeah, why do you say it's not reliable?
because it depends what kind of bees you got in a sample, what type of the day. It's so many factors important, so you can't do a clear test. It's
Yeah.
always change.
Yeah, good answer. So you had some points covered. Anyone else in the audience there in person? Any limitations you think taking one sample and how reliable it is?
I would say anyway, if you see a lot of mites, then that's probably reliable. But if you see no
Yeah.
mites, that may not be.
Okay. Good. Yeah. So what I want you to think is about the spread. I mean, I don't have a great answer for this. Again, apologize. I'm not giving a great answer here, but at least you should think about this next time when you sample for mites. So mites are not equally distributed. Basically, Ella was saying the age of the bees and all that, right? So when you take, so we are trying to be very meticulous. When you do mite sampling, always try to go to the brood area. Don't take mites from some other places, right? Because again, I'm not saying mites are not there in other locations in your hive, but they tend to be more, probably I would say 80 to 90% of the mites are in the brood area where the babies are being raised, right? So because they want to be there closer to that so that they can figure out a larva which is ready for infestation, they go in and start breeding. So if you are focusing on 1 or 2 frames that you want to sample, they should be in the brood area so that your mites. But again, what we have seen in our studies is they're not equally distributed, right? Suppose if I take 200 bees, they're not present on like 100 bees or something. So that distribution has been a really head scratcher for us. I don't have a good way to. So what we did is, I'll just give an example. 4 years or 5 years ago, we did a study. So with about 20 hives. So what we did is you shake 2 frames of bees from the brood area into a Tupperware, and then you mix them really well. And you take 3 scoops. 1 scoop goes into one jar, 1 scoop goes, the second one goes, and all 3 scoops. So what do you think? If the mites are equally distributed, you would imagine that each of those 3 samples I took from those mixed bees, they should be at least close to each other, right? I'm not expecting exact numbers, but at least they should be close by, right? Maybe if I get 10 mites in this jar, I would expect 12 mites in this jar or 15 mites in the third jar, because they're all the same sample from the same hive, from the same bees. They were just taken as 3 different scoops. But you'll be surprised. There is such a huge variation. So I will say, I have some numbers here in front of me as well. So I'll give you one big variation. So in one hive, we got about 29 mites in the first jar, 59 mites in the other one, and 69 mites in the other one. So you see the variation there. So that's what I'm saying. Even commercial beekeepers, I tend to tell them is because for you, you are a backyard beekeeper, probably with 5 hives, 10 hives, 20, I don't know max how many you have. But for a commercial beekeepers, if they are taking they don't have time, right? They have so many hives. So maybe the largest in Oregon has about 10,000 hives. In California, probably someone has 50,000 hives as well. So they don't have time for that many samples to take. So they may take a yard level sample, like 10 samples from this yard. So what I'm saying is that's not a good indication. If you go with that, if suppose I take one sample and I told you, right, I got like 29, 59, and 69. If suppose I took 29, when I took one sample, it was 29. So I'm misrepresenting my numbers, right? And I have a misinterpretation of the entire numbers in the hive. And based on that, if I go with my protocol of my treatments, probably then it's not good. So that's what I want you to be aware. Again, I am not saying this will change your beekeeping at all, but I'm just trying to make sure that you understand the mites are not equally distributed on all the bees. So when you take one sample, it's not a good representation of the number of mites in your hive. Okay, here again for new beekeepers, a strategy, use combination of available mite control. I know as beekeepers, we don't have as many tools as a farmer has for controlling a pest, but whatever we have, I think we are stuck with those. So we have to use them judiciously. So there is a synthetic product called Amitraz. I don't know if anyone uses Amitraz in your group, but we do use at OSU for research and other things. It's the only synthetic product that is available. It still works, but again, we are seeing some resistance being developed in mites for this product. Formic is a very nice product as well. It's the only product that kills mites inside the cells as well. There is no other product that will kill mites inside the cells. So it can penetrate the cells, and there will be some larval damage if you use it the wrong temperatures. So we'll see. So people have documented some queen losses as well, but we haven't seen much of a queen loss. But Formic is a very good product as well if you want to try at the right temperatures. And then oxalic dribble or vapor, those are good options as well. And then I would always encourage you to look for mites after the treatment as well. I know sometimes we assume that it will work for sure, but sometimes I have seen either the product was faulty or something may happen. Maybe the conditions were not right for the product to be very effective. Then you might have lost the chance, right? If you used some miticide in July and then you didn't check until September, probably the damage has been done. See, these mites are notorious. They will transmit viruses so fast that your precious time has gone. So you need to be careful. So I would encourage you to check for mites at least after 4 weeks after treatment to see if the mite levels are at least same or have gone down. So here are again some products listed here. Apivar, Formic Pro, ApiGuard is another nice product. It's a thymol product. And then oxalic is pretty popular now. Hopefully you guys are using oxalic as well. How many of you have tried this one? Varroa, which is an oxalic acid slow release strip. None of you?
I've tried it. Maybe I'm the only one so far.
Okay. I have heard some good things.
Oh,
We
two.
have not tested that product yet, but I think I've spoken to some beekeepers here in Oregon that have used, and I think it's a little expensive, but I think it's a slow release. They have impregnated the oxalic into that. And again, don't think that just putting these strips will take care of your mites. You still have to monitor mites and do other treatments too. But I think what I have heard is it keeps the mite level stable at least throughout the season. And so at least they're not exponentially increasing. And there is another product in the market, probably some of you might have heard it. It's called Vadescana or a double standard RNA. It's called Noroa. Has anyone heard of that? Noroa?
Yes,
No?
I did saw the video about it. Yeah, it's look like good, working well.
Yeah.
I
So
didn't.
Noroa, yeah, it's an RNA. It's a double standard RNA or RNAi, RNA interference. I don't know if you heard of that term before. Uh, it's a good technology. I'm still, I take it with a grain of salt when these products come. So we will test that this year. And maybe if I speak to your group at some other time, maybe I'll give you more data. But it's a very new product. I think some of the bee stores in California, like Man Lake and others, are already selling it. It's expensive for sure. So it's basically the bees have to take this product and then feed it to the larva. Then the mites will feed on the larva, and then that RNAi will go to the mites, and it will impact their gene expression. That is for the gene expression that is responsible for reproduction. I believe those are the genes that they're targeting. So basically, you'll not see a bunch of dead varroa mites, but it impacts the female's reproductive ability. The female will not be able to reproduce. So that's how you decrease the levels. Again, it's not a magic bullet, so you don't have to rely just on that. So you still have to do other things too. But at least I'm glad some new product is coming. But again, I don't have the data on efficacy yet. So hopefully, when we do the study, we'll find out. I think it's already 7:45, so I'll try to go fast on this and then go into nutrition in a minute. So we did some oxalic studies. I think none of you has anyone used oxalic vapor in your group? Okay, so there are a few hands I see. Okay, then I think I'll go over this quickly. Um, so what's the dose of oxalic acid that you guys use for vapor? Like per box. You give me per box. Like you have 2 boxes with brood or 1 box with brood, then how 1 box of brood, like how much is the dose?
Someone in the room said that they used 1 gram, but they're hearing that 4 grams is more appropriate now.
Yeah. So the label has changed. That's another new information I wanted to give your group is so EPA has changed the label now. So you can use 4 grams per brood chamber. So if you have 2 brood chambers with enough brood, then you can go 8 grams total, right? 4 grams is for 1 brood chamber or 1 box with brood in there. So when we were doing this study, it was only still 1 gram. So the label just changed, I think in June, I believe. So now you are legally allowed to use 4 grams per brood chamber. So we were testing all these different doses that I have here. So we published this work. It's open access. So if you are not able to access, email me. I can send you this paper as well. So we tested this oxalic different doses for effectiveness and see if they are also safe for the bees. So these are so we tested different doses. I'm just quickly showing you one study here because of the time's sake. So we tested 3 and 4 because 1 and 2 were not working. That's technically we avoided those doses after the first study and focused on 3 and 4. And 3 and 4, the reason was the 4 had some toxicity to the larva. So we thought 3 would be a little lower dose. And so we wanted to look at those two. So all the parameters I listed there. So we use this system. So here are the results. Again, I'll go quickly here. You can see the 3 and 4 are pretty effective. They're pretty close to each other. And this is something for the new beekeepers I want because I get this question all the time. How long does vapor work, right? How long can you drop mites when you use oxalic vapor? So the answer is only about 24 hours max you get, and then it tapers, but then you're still dropping some mites after 24 hours as well. So, but by end of 48 hours, you see the drop has totally decreased and now it's same as control. So you see 4 grams is that orange one, and this one is your 3 grams. So this is after second application after a week. You still see more mites dropping, right? That's what I was telling you. If you're a new beekeeper, don't assume that one treatment is enough because there are new mites coming every day from those cells. And this is after third application, right? I dropped more mites now. There are 2,000 mites that dropped from these hives. These are averages from about 16 hives. So I'm showing you average, not just from one hive. So you can see the trend here. So that's a take-home message. Older label dosage of 1 gram appears to be inadequate, right? So anyway, the label has changed now. So in our study, 3 and 4 were both effective, but 3 gram was more safer to bees because we saw some larval mortality in the 4 gram dose. Again, the EPA has now approved the label for 4 grams, so you can still use 4 if you want. So a little bit about resistance. I know probably not many of you are using this amitraz, but this is a very important product for the commercial beekeepers. They rely totally on this product, either registered one or there is an off-label use as well for this product. So we wanted to see if there is resistance brewing because some other researchers across the nation have shown that there is some resistance already for this product in those mites that we have. So here we are testing. You see this lower panel here. We have bees with a mite strip attached to the top of this jar, and then you leave those 300 bees with mites in there. And then we see how many mites drop in 3 hours, 6 hours, 12 hours, and 24 hours to see the efficacy. And then we do something called a wild bioassay where we get mites from USDA lab in Baton Rouge as well, because they think their mites have they have more susceptibility because they have not used amitraz for a long time. So we have been getting some mites shipped from there, right? It's funny when you say you can ship mites across state lines. So we are getting these mites on live bees, and then we shake them with powdered sugar, and then we extract them, and then they go into these vials. You see there is a pupa inside this vial, and we have 10 mites inside here. And then we have this jar coated with, all these vials are coated with different concentrations of amitraz, the synthetic product. And then we watch them for 24 hours to see. So this is a more precise way of documenting resistance. So it's still an ongoing study. So hopefully I'll provide more information later. Okay, just for fun, I put something here so that you guys are not tired and sleeping. So how many of you have heard of washboarding behavior before? Okay. Why do bees do washboarding? Just for fun?
Maybe for swarming.
For swarming, okay. Have you seen them swarm after they do this?
Not really, but I'm
Not
just
really. Okay. You're just guessing. Okay. That's a good answer. Okay. So for those who don't know what washboarding is, you see these bees are all lining up like here, right? Their backs are facing up and then their heads are facing down. And I will show you a video if it plays here.
It's their party time.
Party time, huh? Yeah, that's a good joke. Yeah. So, we still don't know. So you still have there's a lot of craze about Nobel Prize these days, right? So you still have a chance to win a Nobel Prize if you want to figure out what these guys are doing. So we still don't know what those bees are doing. As researchers, the only thing we know is these are some middle-aged bees. Middle age means about 14 to 19 days old. That's the only thing we know. Someone has done some research on that. Jeff Pettis, I think that's a researcher if you want to look for that paper. So we still don't know. There are a lot of hypotheses. Some people think they are cleaning their mandibles, but their mandibles are full. They're not even touching the wood, right? Some people think they're cleaning the surface here. There's nothing to clean there. And bees don't care, right? What type of things are outside. They're more clean inside. They don't need to be I mean, this is a nice hive from another beekeeper here in Oregon. So a lot of hypotheses, but still we don't know exam. So the joke was someone said when I told them that these are all middle-aged bees, they said maybe this is a middle-age crisis. Okay, I'll show one more video and then we'll get into nutrition and finish things. Um, Queen dueling. Have you seen queen dueling before?
yes.
Anyone in the audience? Oh, okay. Did you make your queens fight or you saw them inside the hive?
It's different. Sometimes from different hive by accident, queen get into
Okay.
another one, or
Okay.
there is two queen emerge, or
Yeah.
there is a old and mother and daughter. Very rare,
Yeah.
but sometimes
Yeah.
it happens.
Yeah. So for the sake of new beekeepers, so this doesn't happen all the time, right? So during swarming season, when they make a lot of queen cells, right? You'll see probably a dozen queens or more the parent hive will have, and half of the bees with the old queen swarms, and then they make these new queens already, right? So that they will have a queen. So if there are two queens that come out of their cells at the same time, plus or minus few minutes, then they will seek each other and fight for dominance, and one will die eventually because it gets more stings, right? So I'm gonna show you this quick video, but watch for something in this. When I say watch, there is something will come out of the abdomen from the back of one of the queens, and that's the important thing I want you to watch for. What do you think that the material or the liquid that came out of that queen? What do you think that is?
It's great video you have. I'd never seen it.
Yeah, probably this is the only existing video, right? So we
Yeah.
were trying to. So I teach a honeybee. I do a lot of research, but I have a teaching appointment as well. So for my bee biology class that I teach here, we try to make new videos. This was about 15 years ago almost. So we put two queens together that we got from one of your California queen breeders, and then they started fighting, right? So we were probably as a even an experienced beekeeper, you probably heard that the fighting happens only in between virgin queens, right? They're virgin queens that are fighting in the hive. But
No.
these are two mated queens. I put two mated queens and they're fighting. So whenever two queens that are not related also see each other, there is this tendency to fight. So they're trying to sting each other. And so you'll see one of those, I didn't play the video entirely because of time, but so one of them is stung too many times. So that dies and then the darker one survives. So what you saw that spraying, it's called spraying behavior. Like, in your cats and other animals, you see that spraying behavior for territories, but this is not something exactly the same, but it's still called spraying behavior. If you want to read more about it, just Google for spraying behavior. Uh, David Tarpey, he's one of my colleagues in North Carolina State University. So he was at UC Davis when he did his research. So he has published something on spraying behavior. So we know what the material is. So the intent of the queen was to spray the other queen, but this was in a lab setup, so she was not very correct on her target. So when that gets sprayed on the other queen, it's so attractive to the workers, they will come and ball the queen that has been sprayed, and they will probably help the other queen kill this queen. So that's all still a hypothesis. No one really has documented that part, but that's a speculation. But we still know that it's a very attractive compound, and workers get really excited when they see that compound on the other queen or something. So that's what we know. It's called spraying behavior. Uh, so the other topic I chose was bee nutrition. So I'll be quick on this one, just to give some insights to the new beekeepers. What should you think about nutrition, right? I know it's a very broad topic, right? When I say nutrition, see, I put two images here. Nutrition could be your carbohydrates, your honey, right? That's your carbohydrate source. And then pollen is the only protein source for your bees. So nutrition is the first line of defense for like your health, right? If you don't eat nutritious food, then you get compromised health as well. Your immune system is compromised. Same with the bees. It's really, really important for bees to maintain a good immune system because they have so many other pathogens and pests. So what I've done here is micronutrients are on the right side and macro side, right? You all know macronutrients means you need them in bigger amounts. That's probably your carbohydrates and proteins. So we have done a lot of, not just me, I think as a bee research community, people have studied these for almost 100 years about proteins and carbohydrate needs. But what we are missing or we have barely scratched the surface is on the right side, the micronutrients. We still don't know exactly what vitamins bees need, what minerals they need, what lipids, and especially sterols. So that's been one big focus of my lab is looking at or understanding the sterols, right? Like what is your sterol? Cholesterol, right? Humans need cholesterol. Too much cholesterol is bad, but cholesterol is a very important element for your cell membrane development. There are so many functions cholesterol does. It's a source for so many other things. So bees also need a cholesterol, but it's called 24-methylene cholesterol, which is a mouthful. You don't have to worry about it. It comes from the pollen. So the only source for sterol for honeybees is coming from food. They can't make your liver makes cholesterol for you, but bees can't make their own cholesterol. They have to find the cholesterol from outside, from diet, and that comes from pollen. So this is what about a little bit pollen. You get crude protein, amino acids. I'll be quick on this. And nutrition is really important when those bees are being raised. Those called winter bees. I know technically California does not have a real winter, but where you guys live probably. But winter is a really important thing for bees in the temperate climate, right? So we call them diutinous bees. It's a Latin word. Diutinous means long-lived bees. So they have to transition to these bees at some point. So if you ask me in Oregon where I am, probably the bees that are raised in August, September, October, many of them we can call them diutinous bees or winter bees. So those are the bees that are raised to survive the winter. So their physiology is very different. So physiology, when I say physiology, means those bees are not feeding the larva much, right? In August, September, October, at least here, there is less larva, but there is still pollen, decent amount in the hives. The nurse bees, the bees that are 7 to 14 days old, they are the ones eating the protein, right? The foragers that collect pollen foragers, they never touch that pollen, not touching. They don't eat. They collect and deposit, but then they don't eat it. The pollen is eaten by only the young bees, which are about 7 to 14 days old. We call them nurse bees. So those are the bees that need that protein to grow those glands called hypopharyngeal glands and then mandibular glands to produce that food for the larva. The white secretions that they are providing the larva that come from those two exocrine glands that open out from to the mouth, the mandibular glands in the mandibles and the hypopharyngeal glands in the head. So these diutinous bees, the bees that are raised at that point of time, they after they hatch, they're not feeding any more larvae. So whatever protein they are eating, they're storing in their bodies and translating into that fat bodies, right, in the abdomen. So that's a basic change in physiology that happens. And those fat bodies are important for them to survive the entire 4 or 5 months of winter if you live in Minnesota or somewhere more temperate. So that's basically a winter bee. So they're not some magical bees. They look exactly the same as your summer bee, but they have difference, right? You see in this image, the left one, you don't have much fat bodies, but the right one. So this probably you can compare it as a summer bee. This is your winter bee with a lot of fat body stores here. Yeah, I wish they look like this so that you can figure out, right? Okay, so sterols, as I said, are really important for bees. And this is the sterol, 24-methylene cholesterol on the bottom, which is really important for your bees. So insects have to molt. Your honeybee is an insect. So molting means they have to go from one stage to the other, right? And they need these two major hormones called ecdysone and juvenile. So here I have an animation for you to quickly watch to show you the importance of sterols. So the pollen has sterols. They don't look pink. I'm just showing you an animation here that depicts sterols as pink. So bees come and they eat the pollen and they will endogenize this. So they get this endogenous stores now, right? That has gone and they have incorporated into their bodies now. So when they feed that, those sterols will go to the larva as well. See, the larva gets the sterol from the food that the bees have provided. And then the new bee also has some reserves of sterol. But protein supplements, again, don't get me wrong. Protein supplements are fine when you have to use when you don't have enough pollen coming in. But usually protein supplements will not have any sterols. So when your bee feeds on a protein supplement, she doesn't have any of those sterols. So your larva will never grow. It just desiccates. So you can do an experiment like put bees in an enclosure like a flight cage or something and just keep feeding protein supplement. Don't give them any pollen. Those hives will crash after some time. There will be no larva inside those. The bees will start cannibalizing the larva, and then you will see a lot of loss of brood in those hives. So that's why they are very important. So again, if you are interested, you can read. This is another publication we have on 24-methylene cholesterol. We published this in 2019, I believe. Yeah. Uh, so we are doing a lot of those micronutrient studies to understand the importance of these micronutrients, especially sterols. So you may ask, why are we doing that type of work? So it's basically the goal is to improve the existing protein supplements. So if we understand what are the requirements of these, then probably the protein supplements that are in the market, they can be improved with those micronutrients as well. Again, it's a long-term goal. I don't think we are anywhere close, but we need to understand those things as well. So this is the last project, and after this I'll stop. So we got some funding from USDA to look at pollen nutritional composition for pollens, right? So you probably have planted something for bees, or someone has asked you as a beekeeper, what are some good plants we can plant for bees, right? That's a common question these days. We want to improve habitat for bees, but most of the plants are chosen based on attractiveness. If I'm wrong, let me know. See, this is another good habitat for bees, right? So this is my question for you. So if something is attractive, does it mean always good? Sometimes it might be. So if you have this lame example of French fries for you. So if you give me French fries and salad at the same time, I may start eating French fries first probably. That doesn't mean that's a good source of food for me, right? So it's the same again, not a great analogy here, but bear with me. So the question I'm asking is the current basis for selection of plants for bees is based on attractiveness. So someone has looked at these plants and they have seen in a minute there are 100 bees coming to this plant or 200 bees coming on this plant. So we have all that information. I'm not saying that's wrong or not useful. That is useful. But what we want to go is one step ahead of that and see all these plant lists in the NRCS website on USDA, or we have the Xerces Society here in Portland. So they have all these beautiful lists of plants for anyone to choose and plant for bees, right? So what our goal is to have another column here where we test the pollens of all these different species possible, and then give you the data on crude proteins, sterols, amino acids, omega-3, 6, and fatty acids, all those things. Wouldn't that be great? Then you go and say, okay, I have a list here and I choose something for maybe like a Canada goldenrod or something. And then you have all this information and you have additional information on nutrition as well. Only pollen for now. And based on that, you can do a better selection, right? So that's the whole goal, is to have a more judicious selection of plants for planting for bees. So it's a very ambitious project. The funding is done for this already, but we haven't totally finished all samples yet, but we have collected about 100 different species. And collecting pollen is a painful exercise. It looks very simple, right, for some species. So we tried a lot of different techniques, from electric toothbrushes to designing a vacuum to suck pollen from some species. If any of one of you are interested, yeah, let me know. I can give you more information. But this is what we are doing. We're trying to analyze those pollens for all these micronutrients and sterols, both profiles for protein, lipids, and so again, take home message for new beekeepers here. Make sure your bees have adequate pollen throughout the brood rearing season. Pollen is really, really important. Without pollen, your colonies will not grow, right? So I think many of you live in an area where probably there is a lot of pollen coming throughout the season. But if you don't, then you may have to feed them protein supplement at some point of time. But if you choose a protein supplement, I would recommend to buy something that has 5 to 10% pollen mixed in. Because that sterol that I was talking all the time, that sterol is not available in pollens, in the protein supplement. So your pollens have that sterol. So even if you mix a little bit of pollen, you will be well off with your bees. And here are some tips to collect your own pollen if you're interested. I'll send you these slides if you're interested, but you can collect your own pollen, and pollen is good for use even after a year if you store in your freezer, -20 Celsius. Okay. I think there is a publication if you want to read on honeybee nutrition, and I think I'll stop here. And if you still have patience and want to ask questions, I'm still here for another few minutes. Thank you very much.
I had a question.
Yeah, go ahead.
Going back to the Varroa part, when the Varroa are breeding and hatching in the larval cells, you mentioned it was something like one is coming out every X number of days or hours.
30
So
hours.
it
Yeah.
seems to me that there could be like 20 in there, but when I've ever seen them in the larval cells, I only ever see, maybe one or two. So why are there so few,
Yeah.
and what's the record that you've ever seen?
So the ones that you never have seen probably a male mite as well, because they all die. Only the ones that have that have been successfully mated, they are the ones that come out. So the others will die and the bees will remove them. That's why you don't see a bunch of them in the cells when you open, or you might not have seen that many. Is that what your question was?
Yeah, and then what's the most you've ever seen?
Oh, inside, yeah. So, yeah, every 30 hours, but we don't get more than 7 or 8 females they produce inside, and then they have to mate as well, right? Because the female, so there is a competition for mating as well because they have to find the male. So there are some videos if you want to watch. I don't have my own video, but there might be some online where you can watch. Some people have put some cameras painfully behind those cells, and they have shown how a male goes around and tries to find females, and sometimes females go around, and so usually, yeah, in a cell like I showed you that, right? One of those figures that I showed you in that image where I had the entire mite family. So that's what I have seen. Like, but then again, I said there might be 2 or 3 mites get into the same cell. In that case, you may see probably 15 mites probably, but then they're all at different developmental stages. You may see only the 3 female mothers come out along with 3 more babies, I guess. That's a max you can see.
I have a question. Have you done any studies on queen survival? Because they used to say that queens would live 3 to 5 years. And I had a number of years where I would have queens dying frequently. I had 3 different queens in a year. And even if my mite counts were low. So it seems a little better in the last couple of years, but I had years and years and years with low queen survival.
Yeah, no, that's a great question. So we have not done any work on queen survival per se, but that's a concern for many beekeepers. Like we don't have the same survival. I think probably 30 years ago, if you ask beekeepers that have kept that long beekeeping, uh, queens used to survive 4 or 5 years as well. And that's why there is a color code, right? If you know why we paint queens with 5 different colors, because queens used to survive 5 years in the past. That's why the color code came like blue, white, red, yellow, whatever we have. So yeah, these days we are not seeing more than 2 years. I think max I have seen probably in our hives 1 year we intentionally kept the old queens. Probably 3 years, no more than 3 years. So several reasons for those. So the queen breeders are trying their best, but then our bees are in an environment of too much stress, right? There is a lot of maybe sometimes accidentally the insecticides come in. There is a lot of stress on those queens as well, and that's one reason we see it's not about the genetics of the queen, but I think it's more the environmental stressors that are more on the bees these days, and that's probably the reason why we are not seeing that kind of survival that we used to see 30 or 40 years ago.
Okay. Oh, okay.
Go ahead from the floor.
So I'm a brand new beekeeper, but I have a two-part question. One is, how do the mites get originally on a brand new beehive?
Yeah, great question. You have another question, part 2?
Yeah, and then the
Yeah.
second one is, if you're starting a hive from scratch, is there something I can do to like pre-coat my hive, or like should I fume it or spray it or something to like start off the hive
Yeah.
without zero
Without the
mites?
mites? Okay. Yeah, no, very good questions for a new beekeeper. So I'll start with the first one. So the mites are, you can't avoid them, right? So unless you live in an island somewhere without anyone else around there, that's a sad story with mites is mites are there in other colonies around you, right? Depends on where you live. You can have less mites if you're a little more secluded somewhere. So what happens, how bees get mites is even if you just, it's not practically possible. Maybe you started bees this year with zero mites in them, but then they're foraging, right? They're foraging somewhere, maybe 3 miles down somewhere or 2 miles. So mites can be exchanged even when they're foraging on the same flowers. If suppose I have a hive probably pretty close to your location, my bees go to the same foraging arena and your bees come there. So when they're mixing on those flowers, sometimes mites can't fly, but they can jump few centimeters and latch onto the other bee. If there is a mite that falls on that flower accidentally, and your bee comes in, it will latch on, and you need only one single female to get into your hive to get population up. So there is not much a chance of totally avoiding mites. So that's one way you get mites, and then there is a lot of drifting and robbing happens as well. So I don't know if you have heard of that term before. Drifting means if you have some colonies next to you or maybe 10 feet away or something. So especially the drone bees, the males that are produced seasonally, probably in April, May, June in your area, August as well. So they are not very good at coming back to their own hives. So they may drift into the wrong hive. If you have a male drone with a couple of mites on him, if he drifts into your hive instead of some your neighbor's hive, then you get mites as well. So there are ways to get mites is drifting, robbing. If some your bees go and rob another hive which is weak, and they may have a lot of mites and they may get some mites from there. So these are three prime ways: robbing, drifting, and then accidentally getting when they are on the same foraging arena where some other bees with mites are. That's one way because mites can't fly. They have to be accommodated by the bee, right? So that's how they get it. Coming to your second part, as a new beekeeper, the best you can do is maybe I heard the announcements in the beginning that they are going to sell packages or nucleus hives. So when you buy packaged bees, I know those who sell packaged bees also do a good job of mite treatment before they sell you. But again, it's literally impossible to have zero mites. So what you can do is once you start your bees, probably you can do some mite management from day one as well. Like you can do some oxalic or whatever is possible to get the mite levels low. So again, short answer is keeping low mites is the trick. I don't think zero mites is possibility.
I have a question. It's kind of a common knowledge that mites prefer drone cells over worker bee cells, but do you have a statistical data that shows how much they prefer it?
Yeah, no, that's another great question. So, no, I don't recall any studies that have really shown the difference, but you're right. They have preference for drone. Again, as I said in the beginning, uh, the mites have evolved with bees for millions of years now. So they are so good at, uh, so we call it a chiromone, the brood pheromone that is on the larva. So they are mite is a totally different organism, right? But they are able to smell the honeybee pheromone as well. And they are so good, they can tell which one is a drone, which one is a worker, and they can tell the age as well exactly by the pheromone blend that is being emitted. So people have documented, at least I've seen some very not so statistically solid data, but people have shown there is a preference for sure, and they have shown there are more mites in the drone cells when compared to that. But again, I can't answer your second part where to give you an exact data saying there are 50% more mites in drone cells versus, but there is definitely a percentage that goes into drones. And that's why you use that drone traps and all, right? As a backyard beekeeper, you can put a drone comb in that people sell and then make sure that probably many of the mites will go into those drone cells, and then you remove that drone frame at the right time. If you forget, then it's a disaster as well. So
I have a question. Maybe you know, maybe you can guess. Like Apis cerana has a resistance to mites, and mites came from cerana. So how long it may take for Apis mellifera to develop this same protection resistance from mites? How, like evolutionally, how it might take.
Okay. You want me to speculate, so I'll speculate. It can be 20 years, can be 2000 years.
Okay,
So
very good range.
so yeah, it's hard to tell, but it depends, right? Again, because we are giving our bees a lot of protection, right? As a beekeeper. So the reason I said 20 years and 2000 is if you allow all bees to be on their own, don't use any miticide from tomorrow. All beekeepers in the United States, they decide not to use a miticide to help bees, and then probably 95% of the colonies will die, right? And then
No,
whatever
it's
the
not
five,
right. I don't treat my bees and my bees not dying.
so maybe you are in that 5% then, okay? So that's the sad story is the commercial beekeepers cannot do that, right? They will not be
Yeah,
able to survive. So that's
it's
their bread and butter. So they will never get away from that mite regimen they use.
yeah,
So that's the only way you can get faster to it's all about natural selection, right? That's a process, right?
yeah,
Darwin's theory, natural selection is the only way you can get there, and we are not allowing that natural selection to happen quickly because we are helping our bees with miticides. So that's the reason why it might take 2000 years. And if you allow them to die and then you have only few bees, they will get resistance at some point. So that's how the selection pressure. So you have to put that selection pressure on those mites and bees, right? So yeah, I know, great question, but I wish I had a good answer for you.
no, you had a good answer. Thank you.
Okay. Any
In
other
one of the
questions?
last slides, you were talking about or recommending us to collect pollen. I missed what the purpose of the collection is. Do you recommend that we make a pollen patty out of that and feed it back to the bees
Yeah,
somehow?
yeah,
And was
yeah.
that covered in that slide?
Yeah, I can send you the slides to Mike or whoever is in charge of
Great,
collecting
thank you.
slides. Yeah. So the reason I was showing you, I wish I had more time. I just wanted to respect your time, but I put too many slides in there. Uh, so that was to collect your own pollen, because the reason I put that slide a few years ago is someone asked me, can we collect our own pollen rather than buying protein supplements and asking them to have 5% or 10%. So as a backyard beekeepers, yeah, you have the luxury. If you have 5 hives, select the strongest ones. Never trap from weak hives. They will die if you deplete them from pollen. So I have the tips in that, and there is a publication as well. If you want, I can send you that, how to collect pollen. Like, it's a big manuscript that was published a few years ago. So the reason I was showing you that you can collect your own pollen, maybe very small amounts. I'm talking about like maybe a cup, maybe max, and then you add maybe 2 tablespoons in the protein supplement that you will make with your brewer's yeast, or you buy from outside, that's fine too. But I want you to, or you can directly feed them as well. If you think your bees have still some pollen, but you want to add some, then you can make it a small patty form, make it little, add some sugar solution in there, make it a patty, mush it, and then you take a wax paper and put on a wax paper so that it doesn't drop onto the between the seams, and then bees will happily eat that. So that was to collect your own pollen and feed them back. Because if you try to buy pollen from some unknown source, sometimes most of the pollens we see, they are irradiated, because we get this chalk brood and other problems if you're buying from some other beekeeper that you don't know or something like that, because you can transmit some diseases via pollen. That's why if you are collecting from your own hives, you know your hives, and so that's more safety for you to collect your own pollen.
Thank you.
I,
Thank you very
I have a
much.
question on nutrition.
Yeah.
So, you know how microbiome is important in our system?
Mm-hmm.
Mycorrhiza is important in the soil. Have you
Yeah.
have any studies about fungi and mushroom? Fungi mushroom on the bees. Do you have any studies on how mushroom bees fed on mushroom will do better with mites treatment or mite
Yeah,
care?
no, yeah, good question. So I have not done that work, but I can send you a paper or a few papers that I've WSU, our neighboring state, Washington State University. They did some work on mushrooms. So they have extracted mushroom. I think that was one of the. I'm trying to remember the name of the uh, mushroom. It's not coming to me right now, but that's fine. So they have a couple of different mushroom extracts they fed to the bees, and then it was all a laboratory study. And then they fed some viruses to these bees that were fed with the mushroom extract. And then they have shown not mites directly, but they have shown the viruses that the mites are transmitting. They have shown the virus levels have gone down in some of those bees. So there is some work, but again, I have not seen. They were thinking of getting a patent and getting a product for sale as well, which I haven't seen in the last few years. But there is some data that's there that has shown that mushroom extract, some reishi mushroom or something like that, I guess. So they had extracted the compounds from that and fed it to the bees. And then they looked at the virus levels in the bees. And then they saw that the virus levels were going down after feeding the mushroom extract. Great questions. Yeah.
I am wondering, do you have a list of the plants that are very good with pollen? That I know you're talking about getting that on these, more widely produced lists, but do you have any lists of that yourself that you've already done?
So yeah, so if you can wait like another few months, maybe 4 or 5. So we are almost close to getting the data fully. So maybe in the next 5 months, I think we should be able to get at least, we collected like 100 different species of pollen. So we have data for those, not for everything that you might be interested in, but I think we covered most of them, like something like borage, phacelia, and all common things like pear, apple pollen, all the tree fruits, blueberries, like for small fruits. So all cropping systems, plus something that like even maple. Maple is a big thing here in Oregon. And so we have collected maple and whatever is interesting for bees, not the ones the bees don't go. So yeah, email me and maybe in the next few months when it's ready, I can send you the list of those.
That'd be great. Thank you.
I think that the person that you're thinking of for mushrooms and bees and mushrooms and other things too is Paul Stamets.
Yeah. So yeah, he's the guy who is a mushroom guy. And then, but the work was done at the Washington State University Honeybee Lab.
Yes, he worked with someone there.
Yeah. So Steve Shepard is the one that who retired recently from WSU. But so Steve Shepard was doing the experiment, but Paul Stamets was the one that was providing the extracts and was involved in the study as well. Yeah, you're right. Any other questions?
Thank you so much, Doctor. We really appreciate it. It was a wonderful presentation. Thank you.
Yeah, thanks for your interest in bees. Yeah, happy beekeeping. Thank you.
We appreciate it.
Thank you.
Thank you.